Now that we've finished up our investigation of neuroanatomy with sheep brain dissections, you'll be assessed for your understanding with the laboratory practical next week, in addition to an examination on lecture material. With the final laboratory material this week, you'll begin to apply your understanding of brain structures and anatomy to more complex behaviors and manipulations. Some quick other announcements before we get started. Make sure you bring a Scantron for the multiple choice portion of the exam, and as we finish up, start thinking about which brain area you may want to investigate further for your neural circuitry project. Now that we've identified brain regions from the sheep brain dissections, we'll further investigate neuroanatomy from mouse brains, the targeting of brain regions in rodents with stereotactic surgeries, and the scoring of animal behavior in rodent models. The purpose of these activities is to model parallel brain regions, techniques, and behaviors important for investigating human behavior and diseases. And several of these methods have had importance for developing therapies to treat disorders. We left off our investigation of neuroanatomy with the limbic system and its importance for emotion. Some of the most challenging psychiatric diseases to understand and treat are those involving mood disorders, disruptions of emotional behavior. We've now talked about the importance of limbic structures for some emotions, but we didn't really talk in detail about the devastation emotional disorders can induce. I thought we could start today with a famous case study that appeared not too long ago. Most of us are familiar with the actor Robin Williams and his on-screen persona, for the most part joyous, smiling, and goofy. Some would even describe his performances as manic. But many times people didn't see his emotions that were present off the screen. Many times he also suffered from extreme sadness. Indeed, behind his smile and joy was someone in much pain. So how is it that someone who can seem so happy on the screen could be suffering so much? Well, some of Williams' issues with controlling mood can be traced back to his early stand-up career. He started performing stand-up comedy routinely, which caused a lot of stress, and he self-medicated with drugs and alcohol very early on in his career. Throughout the 70s and 80s, he was addicted to cocaine. He struggled with drug dependence even later in his career, developing alcoholism in 2003. Sadly, this depression was accompanied by a form of dementia that left him with anxiety, insomnia, and elevated cortisol levels associated with stress. By the spring of 2014, he had admitted himself into a rehabilitation center for treatment of alcoholism. Shortly after, by the end of 2014, only 66 miles north of where we are now on a foggy summer day, he committed suicide by asphyxiation. Although we now know he had suffered from depression, many people around the world at the time were filled with sadness and questions. How could a man who brought so much joy, who seemed filled with so much optimism and happiness, could have spiraled to this point? There are no easy answers to these questions, but researchers are continuously working on the development of novel therapies that can help treat mood disorders. So how do we address complicated disorders like depression from animal models? Even diagnosing depression in humans can be really difficult sometimes. Often the criteria for diagnosis can be contrasting. For example, with changes in weight or appetite observed as increased weight or decreased weight. There can be changes in sleep, but you can have insomnia or the opposite, hypersomnia. Here's the criteria for diagnosing this depression from the Diagnostic Statistical Manual. I've highlighted a couple of the symptoms at the top in red that I'll be focusing on for the rest of the talk. These include decreased interest or pleasure in most activities, feelings of helplessness or hopelessness, and depressed mood or irritability. These are important behaviors that can be quantified in both humans and animals for psychological study. Diagnosing depression is hard enough, but understanding the brain areas and mechanisms underlying these is an even more challenging task. We can't open up the brains of living people while they're alive and ask them what emotions they're feeling. So, how do we know what's happening in the brain? Because of these restrictions, we commonly rely on animal models to study the neural mechanisms and novel therapies for depression. But you'll notice with this criteria that a lot of it is concerned with feelings or thoughts. When attempting to diagnose a human in the clinical setting, you can at least ask them what they're feeling to find out if some of these symptoms are present. But rodents are commonly used model species, and it's not so easy to discern the emotions from them. Can you tell which one of these mice are depressed? Which ones are sad, angry, scared? You can't talk to the mouse and ask how it's feeling. So how can behavioral scientists model complicated disorders such as depression and anxiety and assess for traits that are common in these disorders? To understand the development of therapies for complicated behaviors, we'll address three questions in this lecture. We'll start by discussing how we can quantify both the behavior and physiological components of emotion in order to study mood disorders. I'll emphasize how we can use animal models to further understand the neural basis for mood disorders to develop novel therapies. In lab, we'll conduct hands-on behavioral scoring and perform mock neural surgeries that can be applied to animal models for developing treatments. We'll finish the lecture by discussing how we verify placement of neural surgeries from rodent microscopy sections that we'll view in the lab. So starting with anxiety-like behavior, many of these behavioral assays take advantage of natural tendencies of rats and mice. For example, pictured here is an elevated plus maze. The physical appearance of this maze resembles its names. It's a plus or T-shaped apparatus that is lifted off the ground. One axis of the T has arms that are enclosed, while the other axis has arms that are open. You'll see from this video captured from an overhead angle that a rodent, in this case a prairie vole here, is first placed into the center of the maze in the open. It briefly explores these open arms, but then fairly quickly retreats into the closed arms of the maze. A standard rodent will be more comfortable with these closed arms where it feels more safe. You can see that once the prairie vole enters the closed arms, it tends to want to stay within them. It peeks out into the open a couple times before retreating back into the closed arms. This test has been used extensively to test the efficacy of anti-anxiety medications. If you give a mouse an anti-anxiety medication, they'll become more adventurous and spend more time in these open arms of the plus maze apparatus. In opposite fashion, the test can be used to test for anxiety-producing effects of drugs or stressful experiences. If a mouse is stressed and has become more anxious, it spends more time within these enclosed arms. Another test for detecting anxiety-like behavior derived from a similar concept is this open field test. Think about a mouse in your house. You may catch it initially out in the open, but as soon as it sees you, it usually runs straight for the walls of the house. If it's out in the open, it risks predation and can be picked off by a hungry owl or an eagle. Rodents will act similarly in this open field test. If they're more anxious, they spend more time along the sides of the arena. If they're less anxious, they'll become more exploratory and investigate the center of the arena. There's two other behaviors to score from this test, and they're auto-grooming and rearing. Like people, animals tend to groom themselves when they are thought to be in a more anxious-like state, and they rear as well, getting up on their two hind paws. During class, you'll score these behaviors in addition to the time in the center squares and time in the outer squares of the maze from a following video that appears sort of like this. So you'll score this video for 3 minutes, and as soon as the animal is placed into the center of the maze, what's important to understand about these behaviors is that they are complicated, and that it doesn't just depend on one brain region. This is also just one component, anxiety-like behavior, that is part of a very complicated set of mood disorders. So how else can we model mood disorders in animals? Similar to measures of anxiety, many behavioral tests for depression-like behavior in rodents take advantage of natural tendencies that they have, and then they look for deviations from the norm. For example, one symptom of major depression that I originally presented was decreased interest or pleasure in activities. Like people, rodents love sugar. So what you can do is test for an anhedonic or a depression-like state in these animals by giving a mouse or a rat a choice between two bottles in its home cage. One bottle just contains plain old tap water. The other bottle has sucrose diluted in water. Animals that are stressed in a depression-like state will show decreased preference for this sugar water compared to the regular water, in comparison to control animals which consume much more of this sucrose water. Another symptom at the top of the list was feelings of helplessness and hopelessness. In rodents, this can be measured using the forced swim test. The test also takes advantage of rodents' natural tendencies. In the forced swim test, the animal is placed in this cylinder of water. Most rodents will naturally swim. However, a rodent that is thought to be in a helpless state will give up and begin floating, a state of immobility. The amount of time that an animal gives up and floats is obtained as a measure of depression-like behavior. I've compiled a video with a journal article and some of my own research that goes into more details about how this test works.
In the forced swim test, mice are placed in a tank filled with water. The mouse remains in the tank for 6 minutes. The subject's behaviors are recorded during this time. At the end of the 6-minute test period, the subject is taken out of the water. After the test is completed, the visual recordings are transferred to a computer or video monitor for analysis.
So here's an example of what these recordings look like. You'll notice that as soon as the animal is placed in the water, it swims vigorously, and most rodents will display this behavior.
An observer measures the time a mouse spends struggling during the last 4 minutes of the 6-minute session. Antidepressant medications increase the amount of time mice spend struggling. This is reflected by shorter periods of immobility time.
From these two tests, a lot of progress has been made for the development of antidepressant pharmaceuticals. Drugs of those classes consistently increase the latency to immobility and decrease the time spent immobile. But how do rodents enter a depression-like state in the first place? Well, it's well established that chronic stress can lead to mood disorders like depression. In particular, we know that from clinical studies that the link between chronic social stress and emotional disorders is well established. Social stressors are something that many people can relate to. You're the new kid at school, and there's a bully who keeps picking on you. Persistent bullying can lead to negative emotions and social withdrawal. But how can you model this in animals? Well, various types of social stressors occur in nature. Lots of animals, like the two male rats that are shown here, are often defending their territories from the constant threat of intruders. This interaction can be reproduced in the laboratory by taking these natural agonistic encounters and applying them in a laboratory setting. This is done by taking a subject and introducing the animal to an aggressive resident's cage. The intruder is at a disadvantage and will almost certainly lose this agonistic interaction. Social avoidance is a common response observed in both humans and other animals following a social stressor. So how do we measure social aversion? Well, following a social stressor, social aversion can be measured using an interaction test. Subjects like this mouse are first acclimated to a large neutral arena with an empty cage placed on one side. During the acclimation phase, the focal mouse displays some moderate interest in the cage. It sniffs it, it passes by, investigates it. Now, let's see what happens when a same-sex, unfamiliar mouse is placed in this mesh cage. You'll see that the focal mouse is captivated by the stimulus. It thoroughly investigates the area around the cage containing the unfamiliar mouse. In fact, in a 3-minute test, most mice will spend an average of around 100 seconds in a tiny zone measuring just 8 centimeters away in each direction around the cage. So this is what a normal mouse does. But what happens when a stressed mouse is placed into this test? Well, many studies researching the effects of social defeat stress have found similar effects. For example, in this experiment with mice that either underwent defeat or control conditions, both groups of mice were first acclimated to the social interaction arena with an empty cage, as I previously mentioned. When durations are scored, no differences are observed in the time spent in the interaction zone between the control and the stressed mice. However, Once a novel same-sex target mouse is placed into this cage, a large difference is observed in the time spent in the interaction zone, with defeated mice spending significantly less time in this zone compared to control mice. Effects of social defeat stress also extend to other traditional assays of depression-like behavior that we previously discussed. In rats exposed to 5 weeks of social defeat stress, you can see that by the second week of stress, the rats begin to show an anhedonic response, less rewarding response to the sucrose solution than they normally would. These rats also show increased immobility during a forced swim test. They give up much easier than control animals in this experiment. But really a great advantage of studying rodent models over humans is that they allow for us to see what's going on underneath the hood, to dive into the brain and discover the neural correlates of these behaviors. We can use these tools to go beyond individual brain regions and discover the underlying neural circuitry. So what are some of the modern tools used to study depression and specific populations of neurons? Well, what we'll be doing in our next lab is using stereotaxic surgery apparatuses to either implant an electrode, which can be used either to stimulate the brain or record from it. And we can also use these surgeries to site-specifically infuse drugs into the brain. Some of my own research has used these approaches to administer SSRI antidepressants, and also site-specific approaches to infuse drugs into particular parts of the brain. Both approaches I found were successful in reversing depression-like behavior. The SSRI approach in particular had some very novel findings. The studies produced some interesting effects both on the brain and behavior. For the chronic administration study, both males and females were stressed and given daily administration of one of 4 doses of an SSRI, a commonly prescribed antidepressant, for 4 weeks straight following social defeat stress. Consistent with the previous study, stressed females spent significantly less time in the interaction zone when compared to control animals. What was interesting about this study was a dose response observed in the female mice. The lowest dose of the antidepressant significantly increased time in the interaction zone, but the effects on social aversion waned at higher doses. I also performed surgeries for site-specific infusions of drug into the brain, aiming specifically for areas of the brain that have connectivity with the amygdala for emotional behavior. We had some infusions that hit the target, and we had some that missed the target with these surgeries, which I was able to determine post-mortem using Nissl-stained sections, similar to what you'll be viewing in our next lab. Infusing the drug into the target area blocked the expression of social aversion in female mice. The drug significantly increased time in the interaction zone when there was a novel mouse in the cage, but it had no effect during the acclimation period when there is no mouse in the cage. In mice in which surgeries missed the target, the drug had no effect on time in the interaction zone, regardless of whether there is a novel mouse present or not. Again, you'll have the opportunity to look at similar types of sections from mouse brains that would be used to confirm these infusion sites, just as I did. In particular, you'll be looking at 2 different types of stains: the Nissl stain, which you previously saw from my research, and the Golgi stain. Nissl first developed his stain in the late 1800s that was based on alcohol and blue dyes. This allowed for visualization, but only of the cell bodies of the neuron. Golgi later created a stain that was derived from silver chromate and shows neurons in their entirety: the dendrites, the soma, the axons, and the cell body. Because of this, Golgi hypothesized that all neurons were connected together in a continuous match, which he named a syncytium. In class, you'll get to view both of these types of staining hands-on with mouse dissections. So you'll get experience performing neural surgeries and viewing sections under the microscope, as you would do to verify placement of neural surgeries in the laboratory. It's going to be a lot of fun, but this is just the neuropharmacological approach. What about the behavioral approach for treating mood disorders? Like animals, we condition ourselves to fear and avoid situations, just like the mice that avoided social interactions. Luckily, unlike animals, we can make ourselves conscious and aware. We can change. We can retrain our brains. And there's now biofeedback therapy that can help us with this task. It does this by increasing awareness of what's going on inside our bodies. A biofeedback machine provides almost immediate output of biological function. Electrode sensors send signals to monitor which display a sound, flash of light, or image that represents your heart and your breathing rate, your blood pressure and muscle activity, and your sweating. There are even recordings, which we're going to use in class, that give you neural feedback from your brain. When you're stressed, your heart rate speeds up, your muscles tighten, and your blood pressure rises. You start to sweat and your breathing quickens. The biofeedback machine allows you to see these stress responses as they occur in real time and get immediate feedback as you try to stop them. It depends on the software, but a lot of times this is done through interactive software where you have to concentrate in order to accomplish a goal, or sync up your breathing rate with software to achieve a task. Over time, some patients can learn to use the feedback to change their physiological responses. You'll get a chance in next class to test out quite a few biofeedback approaches. It's been found that biofeedback and relaxation training, like meditation, are about equally effective in reducing stress and anxiety. But we're not done yet. Both therapies on the behavioral side and the pharmacological side can be continuously be improved, and there are researchers who are working hard on both. With a combination of behavioral therapy and novel medications, we can improve our lives and overall well-being, and may even be able to save a life.